I do understand coming back from vacation. I think we were all a little bit logy today. But then the sun came out and caffeine kicked in. I don't know about you, but I feel a little bit better now. I was really tired this morning. How many of you slept in and then got used to that and then had trouble getting up today? Anybody? Just me? A lot of you? I'm betting. Oh, yeah. Yeah, definitely. Yeah, you get off your schedule and it just messes you right up, doesn't it? Yes. Did somebody have their hand up for a question? Or were you just saying, that's me? I was sleepy this morning. Okay. Whatever. There's always time for questions. Whatever you need to know, you can always chat. Yes, definitely. All right. So what we're going to do is we're going to go into a pear deck. It's another one of those pear decks where all you have to do is participate and show that you're alive and dragging the dots and trying the questions, and you get a 10 out of 10 just for doing that. Before I do that, let me show you also kind of what the bird's eye view of this unit is going to be. I've got my list finally pulled together so you know what's due. The test for this unit is going to be for you guys. The test for your unit is the 10th, which is next Thursday. And I know that comes up a little bit fast, but it's a pretty short practice list, and there's no lab. Okay, there's no lab to do. You don't have to like collect data or graph it or anything like that. We're just going to learn about ATP and cell respiration. And today I'm going to be giving you a lot of direct instruction, and Thursday you're going to be kind of working your own way through some of it after a little bit of guidance in the beginning from me. So you just need to learn about the metabolic pathway of cell respiration, anaerobic and aerobic, and a little bit about ATP and the chemistry of that. And the test next Thursday is going to be a lot like the enzyme test. It's going to be really limited, like 12 or 13 multiple choice questions, and just a 50-point test. So it's not going to be a huge test. But I will tell you that cell respiration is often a topic that students have a little bit of trouble with. So you're going to want to really participate with me when I give you direct instruction and do just the very short list of practice things that I'm setting up for you to do. Okay? So let me show you exactly what this looks like. Right now, today we're doing ATP cycle, but this in yellow is where we are. A bird's eye view of Unit 3. We did the enzyme structure and function unit. We finished with that. Now we're in 3.2, respiration and ATP. So there's some practice and pear decks on ATP and respiration, and then there's a unit test, right? No lab, all right? And that's going to be, we're going to be done with that by the end of next week. And then that very last bit of time that we have before you guys go on vacation is going to be 3.3. We're going to learn a little bit about photosynthesis. It's going to be really self-paced, and then you're going to do instead of a multiple choice test on that, you're going to do a get together with a partner and answer a couple of short essay questions on comparing photosynthesis and respiration. So it's kind of going to feed from this unit that we're doing right here. And then if you understand respiration processes really well, you're not going to have that much trouble understanding photosynthesis to that, because it's the same sorts of reactions, the same things to know about redox and electron flow and where the ATP gets made and where it gets consumed and all of that. So that's where we are. Okay, and then what is this? Okay, so and here's the practice work for this unit here that's due next Friday. All right, your test is the 10th, the other classes are the 11th, your work is due next Friday. This is all practice. Today's pear deck, a little lead puzzle on ATP, it's like 4 minutes long. There's a Google fillable worksheet that's the longest part of this right here, is going step by step through this animation and writing the answers down. And you'll do that on your own, and we'll start doing that next class. And then there's a single little ATP slide that I have little clickable, draggable things that you're going to, it's not real difficult. You can get those, most of this, God, you're done today. And then there's a Google Sheet review of glycolysis that I actually will probably be doing with you. And an ed puzzle on Asian flush, which is actually a condition I thought you might find interesting, and which actually connects to the metabolic pathways of respiration, believe it or not. All of us different races, whatever your ethnicity or your human race is, your ancestry has passed on certain mutations down to you based on what group you're in. You know, if you're Caucasian or you're Chinese or you're Korean or you're African, we all got certain mutations from our ancestors, and every group has its own mutation that kind of got us. We Caucasians have things like Tay-Sachs disease and some other things like that. The Africans have sickle cell anemia that tends to affect them. Caucasians have hemophilia big time. That's what killed the Romanovs. And Asian people tend to have this one gene that makes it difficult to digest alcohol. So that's something that I picked that you can take a look at if you aspire to do that. But today, what we are doing, go back to the plan for Tuesday today, pear duck. So let's get started on that. Let me just find that and get it started. Okay. Even the computer is a little slow today. Okay. So you may remember. But we've already talked about ATP, and ATP is the big energy molecule. We've talked about that before. We're going to go into much more college level detail now. So let me take this and go to presentation screen, joinpd.com, and the little pair says, here's your code. There you go. So you should be able to see that. All right. I'll give you a bit of joining music. I refresh my coffee. Oh. You want it dramatic, Kendall? Here you go. Perfect for the ATP cycle. Absolutely. Be right back. It's so apocalyptic, isn't it? Okay. Is it up on the... Yeah, it's too far away from the end, so I'm just going to... That is kind of cool, though, isn't it? So apocalyptic. Oh. Yes, if 2020 were a song, that would be it. Oh, my goodness. You nailed it. Oh, geez. Whew. And I'm sure Jupiter would be just as similar to that. Okay, so let's get back to business here. Okay, so let's start talking about energy of life. Hum, hum, tum, tum. Okay. All right, so review the energy of life. The living cell is a miniature chemical factory where thousands of reactions occur. You already know that. Remember that metabolism is the totality of all the organism's chemical reactions. A living organism is always in need of useful energy for cell work. Okay, brain. What molecules, plural, okay, have we learned about that are useful for quick energy? I'm wondering what you come up with here. Yeah, carbohydrates and ATP. Yes, exactly. I was kind of hoping that you would cotton on that carbohydrates are also kind of in that group. Yeah, glucose, simple sugars. Oh, those are for quick energy, aren't they? Carbohydrates, sugars, ATP. Yeah, exactly. ATP. And most of you probably said ATP first because I kind of introduced the class as being about. But I wanted to make a point about the fact that we also learned that glucose is, in fact, another quick energy molecule. So good for you for remembering that. All right. So let's just take a look at these two structures here. We've got, I wish that presenting bar would stop. This is glucose down here, right? That's glucose. It's a 6-carbon sugar with hydrogens and oxygens all over it. And then this is ATP. Right? So the structure of ATP is, this is an adenine here, and this is a ribose sugar, and then there's these 3 phosphates. So why is it that they're both, right? They're both defined as being molecules that are really good for use for quick energy. Why is ATP, though, so useful for immediate cell work, and glucose is not? Glucose has to go through thousands of reactions before the energy from it can be released, whereas ATP, immediate, right there when it's needed. So let me tell you why. ATP, most of this molecule, and you can see my arrow moving around it, all of this molecule, all of this part of it right here stays together. It doesn't get broken down by the cell for energy. You're not breaking this bond here, or that bond here, or these bonds here. You're not breaking any of this up, right? The only bond that you're breaking when you are harvesting energy from an ATP molecule, which is what this is, the bond that you're breaking is right here. That's it. You're breaking off a phosphate, and the breaking off of that phosphate, you may recall from your freshman bio and from, I think we've talked about this before, that is what releases energy that can be immediately coupled with some reaction nearby that's endergonic and needs that energy. So this is the only bond that's broken in the ATP molecule when it's being used for useful cell work, immediate cell work, right there using it, right? Glucose, on the other hand, does contain a lot of stored energy, especially the carbon to hydrogen bonds. There's a lot of them in there, okay? So with glucose, you're actually going to wind up breaking every single one of these bonds eventually. If you go all the way through aerobic cellular respiration, all of the bonds in glucose, just about all of the bonds in this glucose molecule, are going to be broken. And the energy from, a lot of times, the energy from the electrons released from those covalent bonds. Remember, covalent bonds are made of shared electrons, right? Every time you break those bonds, some of the energy is collected and then transferred over to be used to help stick the phosphate back on to these guys. Okay? I hope what I was saying was clear there. The rest of this paradigm should hopefully help you understand it. But ATP and glucose, yes, they're both used for energy, but different kinds of energy. ATP can be used immediately for cell work, for little reactions that are going on in the cell that can be coupled with taking this phosphate off, and that energy release is immediately useful for for helping stick an amino acid onto another one in protein synthesis, or for helping actin and myosin fibers pull together during muscle contraction. All kinds of cell work can be helped by pulling the phosphate off of this. But glucose's energy, glucose goes all the way through all the respiration reactions we're going to learn about, and every bond is broken in glucose. And all of that energy in the entire glucose molecule is harvested and transferred to make more of these. Okay, let's keep going. Drag the dot to the molecule that could be used to directly fuel an anabolic reaction. To directly fuel an anabolic reaction or endergonic reaction, right? Direct fuel would be the ATP molecule here. Yes, the red dots go on the ATP molecule because when you take that phosphate off, that energy can immediately be used to do something in the cell to help an anabolic reaction happen. Glucose doesn't work that way. If you break glucose, all you have is a broken glucose. You don't have the type of energy release and the type of loose phosphate that else we're going to talk about that is so useful to get an anabolic reaction to be going. Okay, good job. All right, now drag the dot to the molecule that is completely broken down into carbon dioxide and water by cell respiration, sorry, misspelled, reactions. Right, so now we're on the glucose. The glucose is the food molecule that is totally broken down by aerobic respiration into carbon dioxide and water, right? And the point of doing that is not to make carbon dioxide and water because those are not useful things. Carbon dioxide is waste. Our bodies want to get rid of that. We don't need it, right? So the point is not to make carbon dioxide. The point is not to make more water. You can drink water. You can go to the pond, drink some water, right? The point is to transfer the energy from the bonds in the glucose into sticking phosphates back onto ADP molecules to make ATP molecules. Don't worry, this will make sense the further we go. So how is energy released from ATP? Draw a circle around the part of this molecule that, when removed, can give energy to a nearby process or reaction. This will be a fun thing to look at in terms of all your responses together. Hopefully I didn't give you, try with thinner lines, I think. Otherwise it's going to be really interesting. Really interesting. But that's the fun part anyway, isn't it? Okay, how are we doing? Oh my gosh, you guys are awesome. Yes, that's it. Right there, you're taking the phosphate off. Perfect, perfect. Yes. Oops, where's that silly thing? Here it is. Yes, yes, yes. So it's the terminal phosphate is what you call it. The PO4 group, a polyatomic ion that you learned about in chemistry, right? That's it. You guys have it. I like the pink, right? So remember, not all three, just the last one, okay? Just that last terminal one is the one that comes off. Again, this last one over here is the one that comes off. The closer you get to the ribose, the more strongly held the phosphates are. So those two internal phosphates are actually really hard to get off. That last one is very loose though. Just this only this. The ribose is part of the molecule that just stays put. I like this. Oh, big, thick, blue smiley face. Okay, very good. Okay, you guys got it. Good, good, good. Let's keep going. So I actually showed you this already when we had the enzyme pair deck, but I wanted to just kind of bring it up again because energy coupling is something that you'll see a lot. You'll see that term a lot. And very often when reactions are coupled, One of them is almost always going to be ATP hydrolysis. Look, ATP loses one of its phosphates and becomes ADP, adenosine diphosphate. The energy released from that reaction, which is exergonic, goes to help this endergonic reaction. Putting two amino acids together is an energy requiring process. So if you couple hydrolyzing ATP, which releases energy, with putting two amino acids together, which is an anabolic reaction, which needs energy, and they happen right side by side inside the cell cytoplasm, that's called coupling. Okay. This is the first reaction in the process of glycolysis, which you'll learn about next class. What effect does adding a phosphate to the glucose have on the glucose, do you think, right? So that ATP lost a phosphate. And look where it went. It went right there. What effect do you think that has on that glucose molecule? Think about what glycolysis is. Glyco means sugar. Lysis means to break. The point of glycolysis is to break a sugar. So the first step is sticking a phosphate onto it. What would you guess might be the effect of that? Yes, B is right. It makes it less stable. And as a matter of fact, adding phosphates to things is one of the reasons why ATP is such a good energy stimulating molecule for a lot of chemical reactions. Because a lot of times what happens is the phosphate that comes off of the ATP will attach itself to something, some other molecule that then becomes more activated, less stable, more energized. And that is really the source of the energy that then runs whatever chemical reaction is being helped by ATP. A lot of times it's because the phosphate is activating something once it's loose. ATP drives endergonic reactions by phosphorylation. That's a really important word. Phosphorylation means to stick a phosphate onto something. Phosphorylation, you're sticking a phosphate onto something. The phosphate usually comes from ATP losing a phosphate, and then that loose phosphate sticks somewhere and helps something to happen. Here's a big concept. Phosphorylating a molecule energizes it and often destabilizes it. ATP's value is that phosphate is loosely held and can break off easily, and then reconnect to a nearby substrate or enzyme to activate that molecule. So for example, it's almost kind of like the effect of having a reaction trying to happen without an enzyme, right? So an uncoupled reaction here is a reaction that's trying to have glucose and fructose come together, an endergonic reaction, into a sucrose molecule. And when it's uncoupled, there's no ATP around to help it, right? With the coupled reaction, look at what the ATP does. The phosphate comes off and attaches itself to the glucose. And then the glucose is much more able to be activated and bond to the fructose. It helps the reaction to happen. Okay, so at this point, I think Sal Khan can explain things a little bit better than I can. He's going to explain kind of the more detailed chemistry of this. So I'm going to take a break from the Pear Deck for a moment, and I'm going to share this with you on a different screen. So just stay logged into the Pear Deck, and I'm going to change the focus so you can see this. Well, even the computer is slow today. There we go. Okay, I'm going to come back and share a different screen with you now because you're not going to be able to see that. How are you feeling so far? Does it make sense so far? Can you give me a thumbs up if you're okay so far? Yes? I like the frog, Carter. Great. Okay. All right, good. Excellent. I just want to make sure I'm checking in with you because some of is going to get pretty complicated. I'm going to let Mr. Khan talk about reaction for just a few minutes. Excellent job. Okay, I'm going to turn off my own sound though. Can't hear? Is that what you said? Okay. I think, I mean, it should, if I turn my sound off, that should have helped. Let me, hold on, let me just make this a little bit louder. Okay, let me try that again. Hold on one more minute. I'm still listening. If you still can't hear, I'll come back. How was that? Was that good? Still can't hear. Okay. I'll just leave my mic on then. It should be okay if I leave my mic on.
It's super valuable in biological systems to be able to take a glucose molecule and to phosphorylate it. So let's start with a glucose molecule.
Okay, good. All right. I'm not gonna stop it again. We're just gonna look now.
Phosphorylate, and the reason why is once you have this phosphate group, let me try and put that charge right over there. Once you have this phosphate group, or once you have this negative charge on this glucose 6-phosphate, it becomes much harder for it to leave the cell. The cell wants to hog as many glucose molecules as it can. When the glucose isn't charged, it's able to pass through the cellular membrane, but then once it becomes phosphorylated, it's going to stay in the cell. And glucose 6-phosphate right over here, this is a very important input to a whole series of processes inside cells. Now, unfortunately, this reaction of taking glucose and phosphorylating it, it requires energy. It's enzymatic. It's not going to happen spontaneously. It has a positive delta G. It is endocytic. And so you can imagine what we're going to need to make it happen. We're going to have to use the energy currency of the cell, our good friend ATP. And the way that we're going to make this reaction happen is we're going to couple what's essential to do this hydrolysis of ATP. Exactly why the mechanism. But what's functionally the hydrolysis of ATP into ADP and phosphate, which is very energetic. It is exergonic. It would happen spontaneously under the right conditions. It won't just always happen inside of an aqueous solution.
Okay, so I just wanted to point out there's nothing here you haven't seen before, right? So I came across the top of it, but here's glucose, right? There's glucose. Here is ATP. We were just talking about these two molecules. He's even highlighted the part of the ATP that's going to come off, the phosphate that's going to come off. He's showing you that ATP hydrolysis over here is, I know you can't see it because of the words, but it is exergonic. And we learned what that means, a negative delta G. In fact, it's very exergonic. It's like negative 30 kilojoules per mole versus adding the phosphate to the glucose, which makes unstable, and then there's all kinds of useful things about glucose being unstable and also being stuck inside the cell. You don't want it to leave once you've got it in there. So the cell wants to do this, but you see how it's a bit endergonic to do that. So 13 kilojoules per mole. But you notice how the ATP energy release is greater, negative 30, than the energy required over here for phosphorylating glucose is positive 13. So that's going to come into play later. I'm going to let him keep going. Okay, there's nothing more mysterious about that than the 6 carbon, 1, 2, 3, 4, 5, 6, the 6th carbon of glucose having a phosphate added to it. So glucose 6-phosphate is just glucose with a phosphate stuck to its 6th carbon. And there is an enzyme in here, so you should be familiar with that too. The enzyme's name is hexokinase. Hex meaning 6 carbons, kinase. And any enzyme that is a kinase is an enzyme that sticks a phosphate onto something. That simple.
What's the delta G in this reaction? Well, it's a coupled reaction. View it as a combination of these two reactions. And so, roughly speaking, you can say, well, let's just add the delta G's. So if you add the delta G's here, you're going to get, you add this negative delta G's, exotic, and this positive delta G. You're going to get negative 12.5 plus 13.8. It's going to be negative 16.7 kilojoules. And so this coupled reaction is going to be mixed. It's actually adding as hydrolysis because I'm using some of that energy. But this can happen spontaneously, especially if you can lower the activation energy of the atom. And so let's now look at the mechanism of what happens. Now, without an enzyme, the way that this reaction needs to be is that you have an electron pair over here in this reaction. And it needs to do what's called a nucleophilic attack on this phosphorus. Without enzymes, it's going to be very hard for it to go through the activation region because it's going to be flying around this negative charge from this oxygen. And as electrons don't like going through a negative charge, they're forcing a negative charge. So we need an enzyme to help facilitate this reaction, to help lower the energy to actually start. Essentially, we get these electrons out of the ground. An enzyme, or the term for the enzymes that do this is called hexokinase. And hexokinase. The way it does is it provides ions to, one of the things I want to say, to keep these electrons over here busy. In particular, it has a magnesium ion, right over here. And this is bound to the rest of the hexokinase. This is how I want to mention some hexokinase is kind of right over here. So this can keep these electrons busy. Other ions on the hexokinase can keep these electrons busy. Other ions keep these electrons busy. And so these electrons can sneak in and do the nucleophilic attack. Remember, when we talk about enzymes, they're these proteins we don't see close to hexokinase. These are those complex protein structures right over here, just like this. So you might have the magnesium just right over there, and then maybe the glucose molecule gets bound right here, and then maybe you have your ATP gets bound right over here. Also, just kind of give it exactly, this isn't exactly. But by essentially wrapping it with this positive charge, I'm able to pull the electrons away to help facilitate this nucleophilic attack that needs to happen for the reaction to proceed. So
Again, nothing you haven't seen before. I know this looks complicated, but this is an enzyme, right? Here's your hexokinase. What is that pocket in there called that he's describing? What's the name of the pocket on the enzyme where the substrates fit? What is this here? That opening. Are you saying active? I'm pretty sure you're saying active site. Okay, excellent. That's the active site. And the active site is lined with what kind of charges? Positive or negative charges would be lining that active site. What's he got over here? What's all this? This is the active site stuff here, right? Positive charges. So there'd be magnesium in here, and there'd be other R groups sticking out that would all be positive to neutralize the effect of all these negative charges on those phosphate groups and make it easier to break that phosphate off. In simple language, language, that's what's happening here. And that's really all I wanted you to see from this screen. Every metabolic reaction that you're going to be learning about in glycolysis and the Krebs cycle and all of that, all of those little reactions, those little tiny reactions happening, each one of them has their own enzyme helping it to happen. Each one of them slowly breaks down the glucose. This glucose here gets broken down bit by bit by bit into smaller and smaller pieces. And if you really looked at the detailed chemistry of it, it would look like this. You don't have to know the detailed chemistry of all this, but it's a good thing that you understand that it is there. That in every case, every little step where glucose is broken down or phosphorylated or whatever is happening to it, there's an enzyme environment that's helping that to happen. And a lot of times ATP is involved, either being with losing a phosphate or gaining one. Okay, that's all I wanted you to see with that. Now we're going to go back to the pear deck. Okay, let's go back to the pear deck. There. Okay, let's keep going. All right. All right, so how is ATP hydrolysis used to catalyze other cellular reactions? Now that you've seen that, what do you say? Okay, here you go, take your best guess. Remember, you don't have to be right, just take a guess. And this is straight out of Sal Khan's actual questions after he finished that presentation. The answer is D, guys. The answer is D. Yes. So it's coupled with endergonic reactions. That's the actual answer. It doesn't act like an enzyme catalyst. ATP is not an enzyme. It doesn't have an active site. It can donate a phosphate group to energize something, but it's not going to donate a phosphate group to another ADP. That wouldn't make sense, right? If it's helping a reaction to happen, it's because it's being coupled with an endergonic reaction. It might be activating an enzyme to help that reaction, or it might be attaching to the substrate itself to help that reaction, right? But yeah, that D is the answer. There are probably more specific answers you could say, but D is the best answer here. Good job. Keep going. Here's the structure and hydrolysis of ATP. So ATP stands for adenosine triphosphate. ATP is composed of a ribose sugar here, an adenine here, and 3 phosphate groups. Okay, drag the red dot to the terminal phosphate. Drag the blue dot to the ribose. Drag the green dot to the adenosine. Adenosine, which is actually found in DNA and RNA. It's just being used a little differently here. Okay, all the little dots moving around. Which is which? That's it, okay. So there's the terminal phosphate, there's the ribose, the blue dot goes here, and the green dot goes there, right? So terminal phosphate, not that, those aren't terminal. The phosphate's on the end, right? So the red dot has to go here. This is the ribose, there's the blue dots, and there's the green dots on the adenine. All right, excellent job. We've got the anatomy of the ATP molecule now. Okay. Okay, so this is, Sal Khan was already talking about hydrolysis of ATP. It takes water and it also takes an enzyme to get this terminal phosphate off. But it's kind of neat to see this because this tells you where the different parts of the water go, right? So it's broken right here. If you really look at it, it's broken right here, and then this just remains negative. And the OH from the water goes here, and the other one is just adds to the acidity of the environment. So this is ATP, and this is ADP. adenosine diphosphate, because it only has 2 phosphates. Okay. Energy is released from ATP when the terminal phosphate bond is broken. This release of energy comes from the chemical change to a state of lower free energy and not from the phosphate bonds themselves. And I think Mr. Cant was trying to explain that as well. In the cell, the energy from the exergonic reaction of ATP hydrolysis can be used to drive an endergonic reaction, and overall, the coupled reactions are exergonic. And Mr. Cant showed that really well too when he showed the endergonic positive delta G added to the exergonic negative delta G. When you added them together, the overall reaction was still negative. The delta G was still negative, and therefore it can still go spontaneously if ATP hydrolysis is there. So the 3 types of cellular work, mechanical, transport, and chemical, are powered by this. Can you think of types of cell work that would make use of this? What kinds of things need to happen in a cell, in every living cell? That and be specific as you can, because you can think of some of these things. We've already done some examples. Okay, you're coming up with membrane transport, right? So sodium potassium pump. Remember that when we talked about nerve function? Protein synthesis is another one. Very good. Respiration, the first parts of respiration, yes, but most of respiration doesn't use ATP, actually creates it. Active transport, very good one, yes. Endocytosis, very much so, yes, needs ATP. Yes, maybe some of those endergonic reactions that are required. Yes, membrane transport, active transport, yes. Not really use of food, but yeah, protein synthesis, definitely. What about other things like muscle contraction, right? Within a muscle cell, fibers contracting would definitely require ATP, in fact, physically do. What about sodium potassium pump? What about during mitosis when chromosomes are pulled apart? That's definitely an ATP hydrolysis is needed to have that happen. A flagella beating. There's lots of mitochondria in the base of a flagella of a sperm cell because it takes a lot of ATP to get a flagella to beat. Okay, all those kinds of things are things you need to think about. Almost there. Okay, the regeneration of ATP, right? Once you've broken the ATP off, how do you get it back on, right? You got to put it back on somehow because ATP is so valuable. It can be used up in hundreds of different ways. How do you get the phosphate back on? Well, that's what cell respiration is about. Okay, the energy to phosphate ADP comes most directly from, what do you think? How do you get the phosphate back on once it's been broken off? Cell respiration, that's it. Cell respiration is how you get it back on. Hydrolysis is how you break it off, right? Cell respiration, you're breaking down glucose in food molecules and using that energy to rephosphorylate ADP. That's it. Okay, good. Okay, that's describing the ATP cycle, and I think I'm gonna let you see that because I like that one too. Yes, of course. Sorry. Grammarly again. Okay, here we go. Okay, hold on. I gotta share this with you so you can see it. Okay. Okay, actually, I think you guys need to do it first. Hold on. Okay. Okay. Get up, stretch, take a break. When you come back, we'll finish with the ATP cycle and then you'll be done, okay? Go take a break. Play something inspiring here. Hold on. I got it. Go stretch. Go take a break. You're welcome. Okay, we got to get back to it now. Okay, go over here and so inspiring. Okay, all right. So I actually feel like I can explain this better just on this whiteboard, and it'll be a little bit quicker and a little bit more direct for you if I do it that way. Share my video again. Here we go. Okay. Okay, so again, I am recording all of this, so you can take a look at it later if you need to. But I think, yeah. What I want to do is I want to draw the ATP cycle for you. And when I do this, it will kind of explain, it will tie together everything we've just talked about in the pear deck. And this will also make it really easy for you to go and do the 2 little ATP assignments that are in your classroom. And it be a lot less mysterious if I do this. Okay, so what I'm going to do is I'm going to describe the ATP cycle here with kind of a flow chart drawing. It's going to look a lot like the flow chart that you're going to need to fill in on the Google slide thing, where you're just going to click and drag things around and put them in the right places. So what we'll do is I'm going to put ATP up here on the top. A lot of times what we do is we put adenosine, which is what this is, and 3 phosphates there, and that represents ATP, right? Okay. And then ATP, in order to become, sorry for my arrows, in order for it to become ADP, adenosine diphosphate, ADP, which would be a adenine ribose group with 2 phosphates instead of 3. Right? There. It has to lose, right? It has to lose one of the phosphates. And it also releases energy, right? It releases energy that can be used for cell work. I'll put in all caps, right? And some of the cell work that you guys came up with in that long example when you were in the pear deck and you were thinking about different things that can happen in the cell that would need to have ATP nearby being hydrolyzed and coupled with in order to happen, things like active transport, right? Like the sodium potassium pump. Things like muscle contraction. Muscle fibers, when they're brought together, if you take anatomy with Miss Shamil, she'll go over the sarcomeres with you, right? Where the actin and muscle fibers within a muscle cell, when they're pulled together, it's literally because of the phosphorylation little hooks that are moved from ATP. So muscle contraction is another one. Active transport, muscle contraction, protein synthesis, right? Which is a classic endergonic reaction that requires ATP to put amino acids together, or any other endergonic reactions happening in the cell. Flagella movement, which is kind of like muscle contraction, right? So as a flagella, a beating flagella, it needs ATP. There's a lot in this list. There's a long list of things of cell work that needs the energy released from an ATP molecule in order to happen. Now let's go back over to the other side of this. To put the phosphate back on to ATP, there's a much shorter list of things that allow that phosphate to come back. And instead of this being energy releasing, this is energy consuming, right? So here we have energy is needed. Energy is needed to put the phosphate back on. And where does this come from? Where does that energy, right? Where does the energy ultimately come from to rephosphorylate or stick the phosphate back on ATP? What's the main process, really the only process that does that? Cell respiration. Yes, that's it. Okay, very good. So cell respiration is the main process that does that. And I'm also going to add that there are parts of photosynthesis that also creates ATP in a very similar way that the mitochondria do. So I'm going to put in parentheses photosynthesis can also do this, okay? But photosynthesis doesn't have a net yield of ATP that the rest of the cell use. Photosynthesis only makes enough ATP to help run its own sugar-making reactions. That's all. But it can make some ATP, but only in order to run its own Calvin cycle. Cell respiration, though, is making energy for the whole cell. That's much more generous, right? And ultimately, we need to go back even further. What is cell respiration breaking down that is then being turned into a form of energy that can rephosphorylate the ATP? Ultimately, where is cell respiration getting that energy from to then make ATP? Honestly, it's not that mysterious. Food molecules, guys. Food molecules. Okay? Glucose is the one we're going to talk about the most. But there's also, you can also break down lipids. And if you're really desperate and you're in a starvation mode, your own proteins. Food molecules like glucose and other food molecules can go into their cell respiration pathways and have all their bonds broken down into waste molecules, and then the energy from what was stored in their chemical bonds is transferred into the process of rephosphorylating ATP. Okay, so two more words. Which side? The right side of this? The right side of this? Or the left side of this? Which process is ultimately exergonic? The right side, yes. Okay, so there's another word that you'll need to know. Right, so exergonic is over here. The process of losing the phosphate, that's exergonic, and then that energy goes to do all that cell work, and there's a lot of things, right? That makes this side endergonic, right? Okay, that is the ATP cycle. So now what I'm going to do is I'm going to let you loose to go to get your 2 assignments that you can do for today's practice work. There's an ed puzzle on ATP, the really high energy guy explaining ATP, which I thought was perfect, and I think you'll enjoy that. And then there is another, there is a Google slide where you're going to pull all the players that I just put into place for you and create your own slide of the ATP cycle. Those 2 things. So go get them done now, and then if you feel, that's it. That's it. I'm not going to give you any more to do. And then on Thursday, we'll start talking about glycolysis and anaerobic respiration, which is complicated enough without doing anything beforehand. Okay? Everybody good?